And permitted daily exposure, PDE.
PDE, yeah.
So this is all about like how much of a substance, especially when we're talking about medicines, how much can you be exposed to daily without it causing any problems?
Right. And it gets really interesting when we think about how different medications are made,
Yeah.
often in the same facilities, right? So we're going to look at how this PDE is calculated, making sure your meds are safe, even when it comes to like tiny trace amounts.
Exactly.
We've got a great guide for this, a 2014 guideline from the European Medicines Agency, the EMA.
Ah, the EMA.
We're going deep into the recipe that makes sure your medications are safe.
It's like a recipe, yeah.
Yeah.
We're going to break down each ingredient in the PDE formula and figure out why it matters.
I like that analogy.
So first off, we've got this formula.
Yeah.
What's the basic layout, the structure of this PDE calculation?
All right, so the EMA guideline sets us up with a pretty clear framework.
Oh, good.
The formula goes like this. PDE equals NoAL, that stands for no observed adverse effect level,
Okay.
theta times a weight adjustment. And then we divide all of that by the product of 5 factors, F1, F2, F3, F4, and F5.
5 factors. Okay, that seems,
A lot to unpack.
yeah, we got a lot to unpack there. So let's start with that first part. You mentioned NoAEL.
Right.
So what's that all about?
So NoAEL, the no observed adverse effect level, is basically the highest dose of a substance that doesn't cause any harmful effects in scientific studies.
So it's like the top limit.
Exactly. It's our starting point for determining what's safe.
Okay.
This data usually comes from rigorous toxicology studies, often using animal models.
Makes sense.
Right.
All right, so then we've got this NoAEL,
Yeah.
and then you mentioned multiplying it by a weight adjustment.
Right.
Why do we need to adjust for weight in this equation?
That's a great question.
Yeah.
So you see, the NoAEL we get, it's often derived from studies on animals or even adult humans. But they all have different body weights, you know? So to standardize things for pharmaceutical manufacturing, we usually adjust it to a standard 50-kilogram adult.
A standard 50-kilogram adult.
This gives us a consistent base for our calculations, no matter who was in the initial study, if it was mice or monkeys or a human, you know, who weighs 100 kilograms.
So it levels the playing field.
Exactly.
Okay, so we've got our starting point, our NoAEL adjusted for weight.
Right.
Now we're getting into those 5, the mysterious 5 factors.
Yeah.
F1, F2, F3, F4, F5.
F1 through F5.
Let's start with F1, which you said was species extrapolation.
Right.
And it can range from 2 to 12.
That's a big range.
What's the logic behind that range?
So F1 is where things get really interesting. We often use animal data to predict how humans will respond.
Right.
Which makes sense. We can't be testing, you know, super high doses on people.
Of course, yeah.
But, you know, animals and humans, they're different. They have different biologies, different metabolisms.
Right, right.
So F1 accounts for those differences, how different species absorb, break down, and eliminate substances.
Right.
That range, 2 to 12, reflects how uncertain we are when we translate findings from, say, a rat study to humans.
So a higher F1 means more uncertainty.
Exactly. We're essentially making an educated guess across species.
It's an educated guess, but a necessary one.
And absolutely.
Okay, moving on to F2. This one's set at a fixed 10 for individual variability.
10. Right.
Why 10? Why not 5 or 15?
Well, a factor of 10 for F2 is pretty standard in toxicology.
It's like the magic number.
It kind of is, and there's a good reason for it. We know that even within humans, there's a lot of variation.
Yeah, we're all a little bit different.
Exactly. Our genes, our health, our lifestyles, all of that influences how sensitive we are to different substances.
Right.
So this tenfold factor, it's a safety net to protect even those who might be more sensitive.
It's like a buffer for our unique biology. Okay, so that's F2, individual variability.
Right.
Now we've got F3, another range from 1 to 10. This time it's for study duration.
Study duration.
What does the length of a research study have to do with safety?
Well, think about it this way. If the studies that helped us determine that initial NoAEL were shorter than the potential time someone might be exposed,
I see.
we don't know as much about long-term effects.
Right.
So F3 increases. You could go all the way up to 10 when the study is short. It's like adding a bigger safety net because we don't have all the long-term data.
Longer studies, smaller F3. Makes sense.
Exactly. It shows how important those thorough investigations are.
Yeah, for sure. All right, next up, F4, severe toxicity. Another 1 to 10 range.
Severe toxicity.
When do we need to consider this factor?
So F4 is kind of reserved for those substances that have the potential to cause some serious irreversible damage.
Like the really bad stuff.
Yeah, like cancer or damage to your genes or even your nervous system. If we see that kind of severe toxicity in the studies, then a factor between 1 and 10 might be used to further lower the permissible exposure. It's a reflection of that heightened risk.
That's like an extra layer of caution for the really dangerous substances.
Exactly.
Okay, last but not least, F5.
F5.
Variable factor. And this comes in when a no effect level hasn't been established.
Right.
So what happens when we don't have a clear no effect level?
Well, sometimes the data we have just doesn't give us enough confidence to set a precise NoAEL.
It's like we're missing a piece of the puzzle.
In those cases, we want to be extra careful, right?
Zoris?
So we use F5, a variable factor, usually bigger than 1, to lower the acceptable exposure even further.
It's like we're playing it safe until we get more data.
Exactly. It's a
Yeah.
precautionary approach, acknowledging that we don't know everything.
I like that. It's a humble approach.
Mhm.
So when we take a step back and look at this whole formula, it's like all these F factors, they're like brakes on a car.
Yeah, I see what you mean.
They're progressively making that daily exposure limit more and more conservative.
It's all about minimizing risk.
Right. So we start with NoAL, our basic understanding of safety, adjust for weight to make sure we're comparing apples to apples.
Leveling the playing field.
Exactly. And then each of these F factors comes in, adding a layer of protection, accounting for all those unknowns and potential risks.
You got it. It's a multi-layered approach that really shows how cautious the pharmaceutical manufacturing process is.
It's reassuring to know that they're being so thorough.
Absolutely. It's a systematic way of dealing with the complexities of toxicology and, just the fact that everyone's a little bit different.
So thinking about all those layers of safety built into determining a permitted daily exposure.
Yeah.
What does this tell us about how seriously the pharmaceutical industry takes safety, and how could we apply these same ideas, these layered approaches, in other fields where there's potential risk?
Cleaning validation, especially in facilities making multiple products,
Right.
using the same equipment for different drugs. Our deep dive is going to focus on getting a real grasp of maximum allowable carryover, or MS as it's called, which is central to preventing cross-contamination.
Definitely. Yeah, you've pulled some great material together for this
Right.
that outlines a typical approach to setting these limits, and the MP value is really at the heart of it all.
Okay.
It basically defines how much leftover residue from one product can be left behind in the equipment
Okay.
without posing a risk to patients who take, the next product made in that same equipment.
Okay, so that's the what. But what's the how? How do they come up with that specific limit? What goes into deciding that?
Well, there's a specific calculation involved, and the sources lay out the formula pretty clearly.
Okay.
It boils down to this. MAC equals the permitted daily exposure, and we can call that PDE of the previous product. We'll just call it product A here for clarity.
Okay.
Multiplied by the volume of the first batch of the next product, product B.
Okay.
All of that is then divided by the maximum dose of that second product, product B, and the surface area of the equipment they both came into contact with.
Okay. So that's kind of like the nuts and bolts, right?
That's the core of it.
Yeah, the core of it. But I think, for our listeners, it would be really helpful to kind of unpack each of those pieces, starting
Sure.
with the first one you mentioned, PDE, permitted daily exposure for product A. What does that actually tell us?
Well, think of PDE as like a scientifically determined safety level.
Okay.
It represents the amount of a particular substance that a person can be exposed to every day over a lifetime
Okay.
without any expectation of harmful effects. And these limits are not just arbitrary. They are set by toxicologists based on extensive research.
Oh, wow.
And often they actually build in extra safety factors just to account for any unknowns or variability.
Okay, so that's like the baseline for that first product.
Yeah.
And then, okay, moving on. The next piece of this formula was volume of the first batch of product B. Why is that volume important? Why does that factor in?
Well, because that represents the immediate potential for exposure to that leftover residue.
Okay.
Even a minuscule amount of product A leftover could spread throughout that entire first batch of product B.
Okay.
So by using the minimum batch size in the calculation, it creates like a conservative safety margin.
Oh, I see. So you're really planning for that.
You're accounting for
This is the smallest amount.
the smallest amount.
of that new product that could be affected.
Exactly.
Okay, that makes a lot of sense.
Mm-hmm.
And then the next part of the formula is the maximum dose of product B. How does that play a role?
Well, that takes into account how much of the new medicine the patient will typically be taking, right?
Right.
So if you have a higher dose of product B, that means that even a tiny amount of carryover from product A could result in a more significant exposure for the patient.
So it's not just the amount, it's the concentration, right?
It's the ultimate concentration
Yeah.
that the patient receives.
That the patient receives. That makes sense.
Yeah.
And then finally, surface area of shared equipment.
Yes.
So that's like the part of the machinery that both of those products actually come into contact with.
Precisely. And this is the area where residues from product A could potentially linger,
Okay.
even after cleaning.
Right. Mm.
So a larger surface area obviously means there's more potential for carryover into the next product.
Right. It's like, imagine trying to clean a huge, like a really intricate
Oh, yeah.
piece of equipment. There's just so many nooks and crannies.
More places for things to hide.
Exactly.
Yeah.
So by kind of taking all those pieces into account, the safe exposure level of the first drug, how much of the next drug is being made, the dosage of that, and the size and the shape and all the intricacies of that equipment, you arrive at this MMAC value.
Precisely. And that is the maximum allowable amount of the first drug that can be present in the second drug.
And that's based on science.
It's all grounded in science, and it gives us a way to define what a safe level of cleanliness actually is, quantitatively.
So it's not just like, oh, it looks clean.
It's not subjective.
It's an actual number.
It's a measurable, science-driven limit.
Wow.
Yeah.
And then this MSC number, this is what is used to actually validate cleaning procedures.
Exactly.
So if a company can consistently prove that they are cleaning the equipment to a level below that MSC, they've basically proven that their process is safe.
That's the fundamental principle,
Yeah.
and it provides that health-based criteria for ensuring drug safety in these multi-product facilities.
It's amazing how like something that seems so complex can be broken down into this like logical step-by-step calculation.
Yeah.
I think the big takeaway here is that amicus isn't just some random number. It's a science-driven limit designed to protect patients
Absolutely.
from potential cross-contamination.
Yeah, it really makes you think about how this kind of approach, this risk-based quantitative approach,
Right.
could be applied in other industries as well,
Oh, absolutely.
where even trace amounts of contamination could be a huge issue.
Yeah, it makes you think about like food production or
Absolutely.
cosmetics or
Anything where purity is paramount.
it's all about understanding the risks and then figuring out those limits
Yes.
and setting those clear scientifically backed boundaries.
And having those measurable thresholds.
Right, to manage those risks effectively.
That's it.
That's so interesting.
Yeah. Stage 3 of the cleaning validation life cycle. And honestly, just saying that out loud makes it sound a little dry, but trust me, this is crucial stuff.
It really is, because this is where we prove that the initial cleaning validation isn't just a one-time thing, but something that holds up day after day.
Yeah, if you mess this up, you're risking contamination problems, recalls the whole 9 yards. So let's break it down, starting with the equipment side of things.
Okay, sounds good.
So the documents you shared talk about a matrix or family approach to equipment. Can you explain what that is and why it matters?
Sure. So imagine you have a ton of different tools in a workshop. You wouldn't want to create a separate cleaning procedure for each and every wrench, right?
No way, that sounds like a nightmare.
Exactly. So instead, you group similar wrenches together based on how they're used and the kind of cleaning they need.
Okay, I'm with you.
Then you pick the toughest wrench in that group, the one that's most likely to hold on to residue, and you validate the cleaning process for that one.
So if it works for the worst case wrench, it should work for all the others in that family.
Precisely. It's all about efficiency without sacrificing thoroughness.
Makes sense. Now, the documents also stress the importance of revalidating cleaning procedures periodically, even if they've been working fine for a while. Why is that?
Because things change in a manufacturing environment, right? Equipment gets older. Maybe you switch suppliers for a cleaning agent. Even little tweaks to processes can add up over time.
Oh, I see. So it's like a checkup to make sure everything's still running smoothly.
Exactly. You wouldn't ignore a little crack in your house's foundation, would you?
No way. That's how small problems become huge, expensive problems.
Same idea here. Regular revalidation helps catch those tiny deviations before they turn into major cleaning failures.
And when we revalidate, it's not just about repeating the initial test, right? We're supposed to use historical data too. What does that tell us?
It tells us a lot, actually, by analyzing past cleaning results, we can spot patterns. Are there certain pieces of equipment or product changes that tend to cause more variability in cleaning results?
So we can be proactive and address those potential weak points.
Right. It's all about anticipating problems before they even happen.
I like it. And that leads us to alert or control limits, kind of a technical term. What's the plain English version of what they are and why we need them?
Think of it like driving on a highway. You have those yellow lines on the side, right? They're
Yeah.
there to give you a heads up before you veer off the road.
Right.
Alert limits are kind of like, they're based on your historical cleaning data. So if your current cleaning results start getting close to that yellow line, it's a warning sign.
Time to investigate before you cross the red line and end up with a failed cleaning.
Exactly. It's all about catching those issues early.
Okay, so we've covered the equipment side. Let's talk about the products themselves. The documents talk about a worst-case soilant when evaluating new products. What does that even mean?
So every product leaves some kind of residue behind, right? Some are easy to clean while others are like super stubborn.
That sticky or greasy stuff that just doesn't want to come off.
Yeah, exactly.
Right.
So the worst case soilant is the product that's the absolute toughest to clean. If your cleaning process can handle that, it should be able to handle pretty much anything.
Makes sense. So how do we actually test for this? The documents mention coupon studies and large-scale verification studies. What's the difference?
Coupon studies are like small-scale experiments in a controlled environment. You take tiny pieces of the equipment material, contaminate them with the new product residue, and test different cleaning methods.
So it's like a very focused test.
Right. Large-scale studies, on the other hand, happen on the actual manufacturing equipment after you've run a real batch of the new product.
Okay, so coupon studies tell us about the residue itself, while large-scale studies show us how the whole cleaning process works in the real world.
Exactly. You need both to get a complete picture.
Got it. Now, sometimes you might need to do even more validation studies beyond the routine ones. What would trigger that?
A few things, actually, like say the ingredients in a product change significantly, or you upgrade your cleaning equipment, even if your routine monitoring consistently shows that cleaning effectiveness is dropping.
So basically, any major change or red flag should make you take a closer look.
Exactly. It's better to be safe than sorry.
Okay, final question. Maintaining cleaning parameters in a validated state. What does that actually mean in practice?
It means being super strict about following the established cleaning procedures, the right amount of cleaning agents, the correct temperatures, the specific contact times, all of it.
No cutting corners, no eyeballing it?
Exactly. Any deviation from those validated parameters could mess up the whole cleaning process.
And if you absolutely have to make a change to those parameters, you've got the change control system. Why is that so formal and structured?
Because anytime you change a validated process, you're introducing risk. The change control system helps manage that risk.
How so?
Well, first you have to thoroughly evaluate the potential impact of the change. Then you might need to do some testing to make sure it doesn't mess up the cleaning. And finally, you document and formally approve the whole thing.
So it's about control, accountability, and making sure any changes are made with eyes wide open.
That's the gist of it, yeah.
Has been really insightful.
How consistently we're cleaning, are things really clean? And I see terms like process capability, the CPP and CPK for those kind of quick checks, and then PPPK, process performance, for that long haul view. Now, usually we think of this data fitting a nice bell curve, most results huddled around the average.
Yeah, spot on. Those tools, they work best when our numbers play nice, that normal distribution. Picture this, that bell curve. Classic, right? We're thinking about 68% of our measurements, give or take, landing within one standard deviation from the average. Stretch out to 2 deviations, boom, 95%. 3, over 99.7%. Gives us a good feel for what to expect and helps flag when the process might be veering off course.
So textbook perfect scenario. But, and here's the kicker, your sources, they bring up this challenge with, well, cleaning that actually works is kind of counterintuitive.
All right, here's the thing. When cleaning's super effective, we get a ton of low results, like crazy low, sometimes even undetectable. So instead of that neat bell, our data gets all skewed to one side. It's like a slide with a long tail stretching towards those higher residue levels. That histogram, page 3, nailed it. We're talking lots of squeaky clean surfaces, not so many with much residue hanging around.
Makes sense. But now, hold on. All those go-to statistical tools, those control limits, and the rules for spotting trends, like Western Electric, they're built on that bell curve assumption. So cleaning data goes rogue, doesn't fit the mold. Big question then, how do we even use these statistics to validate and keep tabs on cleaning? Our mission, should we choose to accept it?
This is where it gets tricky, right? Can we twist our stats approach? Your material mentions trying to, well, force the data into that normal distribution, like math wrestling it. Page 5 talks about Box-Cox transformation.
Oh yeah, that equation. Y transformed equals, hold on, y original to the power of lambda minus 1, all divided by lambda. Oof, my brain hurts. And from what I see here, your sources are hinting this might not always be the magic bullet for cleaning data.
Exactly. Even with that transformation voodoo, the data from a really effective cleaning process, it often still refuses to play by the normal distribution rules, or even fit into other common distributions, to be honest. The Anderson-Darling test results, page 9, they made that pretty clear. I mean, so many zero or near zero readings, it's tough to cram it into those standard molds.
So if forcing our data into that bell curve is a no-go, what's the alternative? Aha, the empirical distribution function. This is where your material gets kind of cool, a more data-driven approach.
Absolutely. We ditch those theoretical distributions and just embrace the chaos, the actual distribution of what we see in our data. Percentiles, the 99th, the 99.9th, those become our guides for setting those practical control and alert limits. The example on page 13, remember, they went with the control limit of 99.5 day 25 centimeter soars, shows how we use the data itself to define what's acceptable. It's like saying, hey, looking back, 99.5 D 25 centimeter soars, that's a level we hardly ever bust when things are running smoothly.
Gotcha. So we're setting these limits based on reality, not theory. How does that change our day-to-day monitoring, though?
Good question. It's all about tracking those results against those empirical limits. Page 13 breaks it down. We stick to those original acceptance criteria, sure, but now we're also on the lookout for trends, how things are moving relative to that performance control limit. If data points are creeping up on or, crossing that line, it's a red flag, time to investigate. We might even tweak those trending rules like, hey, 1 point over the control limit, let's review.
More flexible, more in tune with the actual cleaning data, not bound by those normal distribution rules. And this ties into, I see it here on page 18, this whole idea of cleaning as a continuous process, right? Always monitoring, always evaluating.
Couldn't agree more. Maintaining truly effective cleaning, it's way beyond passing some initial test. It's about keeping things consistently clean throughout the whole process lifecycle. And that means learning from the data we gather, even when it throws those textbook statistical models out the window.
And that leads me to a final thought for everyone listening. If we think about it, tons of real-world processes, they generate data that's, well, messy, not perfectly normal. So what other fields could benefit from loosening that grip on those traditional statistical assumptions? Maybe embracing those more empirical, data-driven ways of setting performance limits. Lots to chew on there. Thanks for joining us for this deep dive.